Good morning, children. Work hard, dream big, never give up. Before going to the lesson organization of tissues, let us see the recap. Types of cell division. Three types of cell division: amitosis, mitosis, and meiosis. Previous class we discussed only amitosis and mitosis. Okay, children. Now we pass on to meiosis. Meiosis, in mitosis as well as in meiosis, all the four phases are same: prophase, metaphase, anaphase, and telophase. Before going to mitosis, let us see some words, some terms. You should be familiar with the terms. Haploid. What do you mean by haploid? Single set of chromosomes. Diploid. Two complete set of chromosomes. Then heterotypic division and homotypic division. Heterotypic division means heterotypic division or first meiotic division. Children, why it is called heterotypic division? The daughter cells resulting from this division are different from the parent cell in the chromosome number. Homotypic division means homo means similar. The daughter cells are similar to the parent cell in the chromosome number. And now let us pass on to meiosis. The term meiosis was coined by Farmer in 1905. Meiosis is the kind of cell division that produces the sex cells or the gametes. It is also called reduction division because the chromosome number is reduced to haploid from diploid, that is n from 2n. Meiosis produces 4 daughter cells from the parent cell. Meiosis consists of 2 divisions. They are heterotypic division or first meiotic division, then homotypic division or second meiotic division. Now, heterotypic division. It divides diploid cell into 2 haploid cells. The daughter cells resulting from this division are different from the parent cell in the chromosome number. It is called heterotypic division. This consists of 5 stages. What are the 5 stages? Prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. Children, now we pass on to prophase 1. Prophase 1 takes a longer duration and it is subdivided into 5 stages. They are leptotene, zygotene, pachytene, diplotene, and diakinesis. Now let us pass on to leptotene. The chromosome become uncoiled and assume long thread-like structures and take up a specific orientation inside the nucleus. They form the bouquet stage. See, usually the chromatin reticulum uncoiled and the thread become a thread-like structure, and this is leptotene. Then zygotene. Zygon means adjoining. 2 homologous chromosomes approach each other and begin to pair. Pairing of homologous chromosome is called synapsis. So what do you mean by synapsis? Pairing of homologous chromosomes is called synapsis. Then pachytene. Pachytene is also a little longer stage. The chromosomes are visible as long paired twisted threads. The pairs so formed are called bivalents. What do you mean by bivalents? The pairs formed between the chromosomes are called bivalents. Each bivalent now contains 4 chromatids. This stage is called a tetrad stage. What do you mean by tetrad stage? Each bivalent contains 4 chromatids. Homologous chromosomes of each pair begin to separate. What do you mean by homologous chromosome? Homologous chromosomes means the associate chromosomes are called homologous chromosomes. If in a pair 2 chromosomes are there, so the other chromosome is called homologous chromosome. They do not completely separate, but remain attached together at one or more points by X-shaped arrangement known as chiasmata. So what is chiasmata? The homologous chromosomes, they do not completely separate but remain attached together at one or more points by X-shaped arrangement. This is called chiasmata. The chromatids break at these points and the broken segments may be interchanged. This process is called crossing over. So, the exchange of segment is called crossing over. So, as a result, genetic recombination takes place. Now, we pass on to diplotin. Each individual chromosome of each bivalent begins to split longitudinally into two similar chromatids. The homologous chromosome repel each other and separate. Then the chiasmata begin to move along the length of the chromosome from the centromere towards the end, resulting terminalization. Then we pass on to diakinesis. The paired chromosomes are shortened and thickened. The nuclear membrane and the nucleolus begin to disappear. Spindle fibers also reappear or make their appearance. Then we pass on to metaphase 1. The chromosomes move towards the equator. Equator means the center place. And finally, they orient themselves on the equator. The two chromatids of each chromosome do not separate, so the centromere also does not divide. Now let us pass on to anaphase 1. Each homologous chromosome with its two chromatids and undivided centromere move towards the opposite poles of the cell. This stage of the chromosome is called dyad, D-I-A-D, dyad. Then telophase 1. The haploid number of chromosomes after reaching their respective poles become uncoiled and elongated. So the nuclear membrane and the nucleolus reappear and thus two daughter nuclei are formed. Then cytokinesis 1. What is cytokinesis? Division of cytoplasm is called cytokinesis. So the cytoplasmic division occurs and the two haploid cells are formed. Children, now we pass on to homotypic division. I told you what do you mean by homotypic. It is similar to the parent cell in the chromosome number. So in this division, two haploid cells are formed during this first meiotic division, divide into four haploid cells. The daughter cells are similar to the parent cell in the chromosome number. It consists of 5 stages, namely prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. Prophase 2. The centriole divides into two. Each one moves to the opposite poles. Then the asters and the spindle fibers appear. Then nuclear membrane and nucleolus disappear. Then metaphase 2. The chromosomes get arranged on the equator. The two chromatids are separated. Then anaphase 2. The separated chromatids become daughter chromosome and moves to opposite poles. Then telophase 2. Then the daughter chromosomes are arranged in the center. The nuclear membrane and the nucleolus appear. Then cytokinesis 2. Two cells are formed from the each haploid daughter cell, resulting in the formation of 4 cells with a haploid number of chromosomes. Children, now we pass on to significance of meiosis. The constant number of chromosomes in a given species is maintained by meiotic division. This is the significance of meiosis. Now let us pass on to differences between mitosis and meiosis. Mitosis, it occurs only in somatic cells. Meiosis occurs in reproductive cells. And mitosis is involved in growth and occurs continuously throughout the life because it related only through somatic cells. And meiosis involved in gametic formation only during reproductively active stage. So meiosis occurs in the reproductive cells. Then mitosis, it consists of only single division, but meiosis consists of two division, namely meiosis 1 and meiosis 2. Two diploid daughter cells are formed in mitosis, and meiosis at the end 4 haploid daughter cells are formed. Then the chromosome number in the daughter cell is similar to that of the parent cell, that is diploid in mitosis. But in meiosis, the chromosome number in the daughter cell is just half, that is n number of chromosome, haploid number of chromosome in the parent cell. Then the last point, identical daughter cells are formed during mitosis, but in meiosis, daughter cells are not similar to the parent cell and are randomly assorted. Now we come to the end of the class, children. Now I am going to give some questions as homework. What are the questions? I am going to give 4 questions. List out the differences between mitosis and meiosis. Second question, why should gametes be produced by meiosis during sexual reproduction? Third question, what is tetrad? Fourth question, what do you mean by synapses? Okay, thank you, children.